We are taught from childhood that vitamin A is the eye vitamin. Eat your carrots, improve your night vision. Yet at the microscopic level, this molecule actively regulates cellular differentiation across the entire body. The ancient Egyptians observed this long before biochemistry existed. They recorded in hieratic script their practice of feeding raw liver to patients to cure blindness. Without knowing it, they were delivering massive doses of a stored, active molecule straight from the organ that processes it. The biochemical family responsible for these effects is collectively known as the retinoids. We classify them into three distinct active forms: retinol, retinal, and retinoic acid. Tracing this molecule's physiological life cycle explains exactly how it dictates normal human health and why its deficiency causes systemic breakdowns. It also reveals how a massive dose provides a targeted cure for a specific deadly leukemia. Ultimately, retinoids serve as active structural architects, dictating the fundamental integrity of human tissue at the genomic level. This schematic illustrates the dietary pathway of retinoids. Because vitamin A is fat-soluble, its absorption in the gut relies strictly on a robust pancreas secreting lipases and a functional liver supplying bile salts to break down the lipid carriers. This means a patient can eat a perfect diet and still suffer severe deficiency. Malabsorption syndromes like cystic fibrosis or celiac disease disrupt lipid digestion. The vitamin never crosses the mucosal wall of the gut. Once it reaches the liver for storage, it faces another transport constraint. Fat and water do not mix, meaning retinol cannot circulate freely in plasma. To exit the liver and reach peripheral tissues, it binds to two specific carrier proteins: retinol-binding protein and transthyretin. Transthyretin also serves as a major precursor protein for amyloid formation in pathology, directly tying the retinoid transport system to broader systemic diseases. A patient's systemic vitamin A status relies entirely on their internal anatomy. You need an intact gut, a functioning pancreas, and a healthy liver to extract and transport the molecule. Once inside the target cell, the molecule must be activated through a precise oxidation cascade. Retinol, an alcohol, is converted into retinal, an aldehyde. That retinal is then oxidized into retinoic acid, a carboxylic acid. If this sequence sounds familiar, it mirrors standard ethanol metabolism. The exact same classes of enzymes drive both processes. Alcohol dehydrogenase handles the first step, and aldehyde dehydrogenase handles the second. This schematic shows the structure of those dehydrogenase enzymes. They share a critical vulnerability. Both require a zinc ion as a mandatory cofactor. Until that zinc locks into place, the enzyme cannot activate the oxidation process. If a patient has a severe zinc deficiency, such as in acrodermatitis enteropathica, the entire cascade freezes. The enzymes fail. The cell is stuck with inactive retinol and cannot produce retinoic acid. The patient will present with severe skin inflammation and failed epithelialization. A trace mineral deficiency masquerades perfectly as a severe vitamin deficiency at the molecular level. When successfully activated, the molecule diverges into two different functional paths. In the eye, the aldehyde form, retinol, binds to the protein opsin. This forms rhodopsin, the core light-sensitive pigment that allows the retina's rod cells to perceive night vision. The systemic path works entirely differently. The final carboxylic form, retinoic acid, is highly lipophilic. It bypasses the cellular membrane, enters the cytoplasm, and dives straight into the nucleus. Inside the nucleus, it binds to specific zinc finger receptors. These are linked to retinoid X receptor, or RXR, enhancer elements positioned directly on the DNA sequence. The retina uses the molecule strictly for phototransduction to interpret light. The rest of the body uses it to directly modulate active gene expression, telling cells exactly how to grow and specialize. Remove retinoic acid and normal cellular differentiation fails. The tissue defaults to a hardened, abnormal state, triggering widespread squamous cell metaplasia. In the eye, this manifests as conjunctival xerosis, extreme dryness, which rapidly degenerates into severe corneal ulcerations and keratomalacia. On a physical exam, this localized ocular metaplasia produces distinct foamy white deposits on the temporal side of the sclera, known as Bitos spots. On the skin, the lack of proper epithelialization leads to extreme dryness and follicular hyperkeratosis, a condition called phrenoderma. This breakdown extends inward. The mucosal epithelium of the respiratory and gastrointestinal tracts loses its structural integrity. The patient's first line of immune defense collapses, leading straight to recurrent pneumonias and chronic diarrhea. The opposite extreme is equally dangerous. Because the molecule is fat-soluble, the body cannot efficiently excrete excess amounts in urine. Overdose leads directly to toxic accumulation, known as hypervitaminosis A. Acute toxicity causes sudden intracranial hypertension. The patient presents with a severe headache, nausea, and blurry vision, a clinical picture that perfectly mimics a brain tumor. Chronic toxicity slowly poisons the system, resulting in unexplained weight loss, severe hepatotoxicity, and an increase in bone fractures, as normal osteoblastic activity is suppressed. Clinical retinoid pathology is an extreme pendulum. On one side, a total structural collapse of the human epithelium, on the other, systemic toxic overload. We can take this molecular model and apply it directly to oncology, specifically to treat a highly lethal blood cancer, acute promyelocytic leukemia, or APML. APML is driven by a specific genetic error. It stems from a translocation between chromosome 15 and chromosome 17. This fusion creates the PML-RAR alpha gene. It produces a mutated receptor protein that physically clamps down on the cell's DNA. The primitive promyelocytes are trapped in an immature state. They cannot mature into functional white blood cells. The solution relies on overriding this mutated receptor by flooding the system with massive pharmacological doses of all-trans retinoic acid, ATRA. The sheer volume of the vitamin forces its way into the defective receptor, shattering the genetic blockade and forcing the leukemic cells to finally mature. This rapid success creates a new problem. The sudden mass differentiation of thousands of white blood cells triggers a massive inflammatory response known as differentiation syndrome, leading to fever, pleural effusions, and acute respiratory distress. To prevent the patient from drowning in their own inflammatory fluid, this syndrome requires immediate rescue therapy with high-dose dexamethasone to suppress the reaction. A molecule normally required to prevent cellular dysplasia is weaponized. We use it to force terminal differentiation and kill cancer cells. Vitamin A is a molecular architect operating directly on the human genome. The cystic fibrosis patient who suddenly loses their night vision, the child with severe hyperkeratosis, and the leukemia patient entering rapid remission are all experiencing different points along the exact same biochemical axis. Their clinical outcomes are strictly dictated by how efficiently the oxidation pathway converts the raw material, and how successfully that active molecule binds to the nuclear receptors. By mastering the first principles of cellular metabolism, pathology stops being a disconnected list of symptoms. It becomes a predictable sequence of molecular logic.